Welcome to the Predicine Liquid Biopsy Symposium. My name is Shidong Jia, founder and CEO at Predicine. Joining me today is Dr. Richard Wenstrup, Chief Medical Officer at Epic Science. I would like to start today's talk with an exciting announcement. Predicine and Epic Science has entered into a global strategic partnership to offer biopharmaceutical company access to Predicine's ctDNA-based testing and Epic's CTC-based testing. Through this partnership, together, we provide a first and best-in-class, a most comprehensive liquid biopsy biomarker solution to biopharma partners. Speaking about biomarker, tissue is the gold standard in clinical practice. However, tissue is often the issue. Tissue biopsy is invasive, hard to access, expensive, and not feasible for longitudinal study. Recent advances in science and technology have made blood as the ideal source for biomarker studies. Peripheral blood is rich in biomaterials, for instance, circulating tumor cells, and circulating DNA shedding from tumor tissue, CTC, and normal cells. Blood as a liquid biopsy is non-invasive, easy to access, affordable, and suitable for longitudinal study. On the right side, blood provides a valuable source for DNA-based genotypic insights and CTC-based phenotypic insights. So for instance, here we show the ctDNA-based molecular profiling of important cancer genes such as EGFR, HER2, PIK3CA. And CTC showed its value in CTC-based staining for PD-L1, HER2, and androgen receptor. Through this multi-technology approach, CTC and ctDNA analysis answer different questions, but they are completely complementary to each other and definitely not competing. This is the rationale that brings Predicine and Epic Science together and to form a global partnership in combining CTC and ctDNA for biomarker study. Based on our own experience in drug discovery, disease biology, and clinical biomarker development, we published a prospective review article illustrating the future need of comprehensive biomarker profiling. ctDNA and CTC are the main topic of today's discussion. Speaking about ctDNA, Predicine had developed its proprietary liquid biopsy NGS platform, enabling deep coverage of a wide range of common cancer variants. It detects SNV, indel, fusion, CNV, TMB, MSI, among others. As a differentiator, Predicine ctDNA technology can detect cell-free DNA-based gene deletion, for instance, the biallelic deletion of BRCA gene or PTEN, p53, or RB tumor suppressor gene. Besides DNA, Predicine, the cell-free RNA technology, can detect RNA splicing and novel fusion. Today, Predicine liquid biopsy assay has been widely used by leading global pharma in clinical study, ranging from early cancer detection and intervention to localized and metastatic disease and to resistant mechanism and recurrence detection. Beyond ctDNA alone, here I would like to share with you our recent collaboration study with Dr. Massimo Cristofanilli at Northwestern University, demonstrating the association of Predicine ctDNA NGS platform with the CTC and CTC cluster in metastatic breast cancer. Specifically, the red figure on the top is showed a higher number of CTC was associated with genomic alteration in breast cancer genes such as ESR1 or GATA3. The lower figure and showed the genomic alteration in CDH1 and CCND1 associated with a higher number of CTC. Of note, the CDH1 gene is involved in the production of E-cadherin, a gene that plays an important role in cell adhesion, chemical signaling, cell movement, all of which may contribute to the metastatic process. Collectively, these ctDNA and CTC studies demonstrate the complementary nature of ctDNA and CTC for comprehensive biomarker profiling, and also provide important implications for clinical management of cancer patients. I would like to pause here and pass the microphone to Dr. Wenstrup to introduce EPIC's CTC capability.
Thanks, Dr. Gia. Here on this slide, we outline the EPIC Sciences CTC platform. This rare cell detection platform is a spin-out from the Scripps lab by our founder, Dr. Peter Kuhn. That's Dr. Kuhn there on the panel on the left. He's now at USC, and we continue to work closely with him in his capacity there. The technology provides best-in-class limit of detection, 1 in 10 million cells. We call that no cell left behind. That is an actual laboratory slide there at the top, on which there are approximately 3 million cells deposited on the slide, and there is a single red circulating tumor cell, or CTC. We define a CTC as a carcinoma cancer cell found in the peripheral blood that has an intact nucleus, is positive for cytokeratin, negative for CD45, and has morphological features associated with cancer. On the second panel from the left, when a blood sample arrives at our lab and the blood is spun down, the red cells are lysed, and then through a proprietary cell deposition process, the white blood cells and the CTCs are deposited on the slide, allowing for visualization of all 3 million cells. Now that we have moved to comprehensive cancer profiling, the process also includes preserving the plasma for ctDNA analysis. On the third panel from the left, the staining process includes our use of molecular markers unique to white blood cells and CTCs, allowing us to differentially identify each. The final step in the process is the use of high-resolution image analysis using SICE technology. An image gallery is created. I would also note that XY coordinates for each of the 3 million cells are made, allowing us to go back and analyze spatial, morphological, cellular, and genomic features. These features include genomic instability, cellular heterogeneity, and neuroendocrine-like features. We can also pick individual CTCs for further analysis, such as NGS genomics. I will also point out that staining and CTC genomic analysis can take place on these cells after they've been stored for many years after plating. About a year ago, we validated a process whereby we isolate plasma for ctDNA analysis from our Streck tubes without adversely affecting our CTC preparation process from the Buffy coat, as shown here. Without that process, we wouldn't be able to do this joint presentation today with Predicine. This slide shows the power of the Predicine and EPIC Sciences partnership. It's multi-technology testing of CTCs and ctDNA from a single blood draw. Using the analytes from the tube shown on the left, ctDNA, CTCs, and white blood cells, we can perform CTC enumeration, sequencing from ctDNA through our Predicine partner, rare cell detection, single-cell genomics, and phenotype and clonal characterization, and immune cell characterization. So from this blood draw, there's a massive amount of information, and while ctDNA companies can provide answers to certain questions, EPIC's platform, combined with Predicine, can provide more complete answers. For example, enumeration of CTCs in metastatic breast cancer can provide a gauge of the patient's prognosis and monitoring response. We can confirm that newly suspected cancer is metastatic disease and not a new primary cancer. We can provide evidence of resistance to other therapies through further treatment lines. We can provide important information on immune status, not only to test for IO targets like PD-L1, but also to understand that patient's T cells are activated, looking at CD4-8 ratios, or exhausted, etc. ctDNA analysis of the PIK3 kinase status determines if alpelisib is a treatment option. And continued characterization of the protein expression as the disease evolves will determine when new treatment will be needed and what treatment would be effective. This slide demonstrates that comprehensive cancer profiling requires contributions from both circulating tumor cells and circulating DNA. The added value of these two technologies is shown in three different contexts here on the slide. At the top, in blue, in prostate cancer, CTCs on the left exhibit important protein markers such as ARV7, AR protein, PSMA, SLFN11, and PTEN. Complementary CTD DNA analysis on the right shows analysis of the AR and PTEN loci, PIK3 kinase, and BRCA genes. For breast cancer in pink, on the left, protein analysis demonstrates HER2, ER, and PR status. On the right, using Predicine's ctDNA, ERBB2, ESR1, PIK3 kinase, and BRCA status is identified. CCP also provides pharma partners a wide range of immunofluorescence markers on the bottom left and genomic markers on the bottom right. The figures shown on this slide, taken from the recent paper by Radovich et al. In JAMA Oncology, show the combined power of CTC analysis and ctDNA analysis. The ctDNA analysis in this slide was a tumor fraction assay. This was a phase 2 multi-center randomized clinical trial with 196 triple-negative breast cancer patients. This study investigated the association of ctDNA and CTCs after neoadjuvant chemo with disease recurrence in triple-negative patients. Among the 196 female patients, detection of ctDNA was significantly associated with inferior disease-free survival. The combination of ctDNA and CTCs provided additional information for increased sensitivity and discriminatory capacity. In the Kaplan-Meier on the left, the best survival was from patients who had no CTCs detected and no ctDNA detected, while the worst survival were in patients who had both CTCs and ctDNA detected. Similar trends were observed for both disease-free survival and overall survival. So this study demonstrated the clinical utility of a combined ctDNA and CTC approach in breast cancer.
Thank you, Dr. Wenstrup. Predicine and EPIC has established a global laboratory network. We have CLIA and CAP laboratory set up in California, USA, Shanghai in China, Geneva in Europe, and Singapore in Southern Asia. Through our global QMS system and identical SOP, we provide a harmonized assay in US, EU, and China to support global clinical trials. In clinical study, blood sample collected at the site will be sent to the lab of EPIC Science first for CTC isolation and plasma preparation. ctDNA molecular profiling will be conducted at the Predicine lab, and CTC phenotypic profiling will be conducted at the EPIC lab, with a combo CTC and ctDNA result sent back to the pharma clients. By this way, our integrated ctDNA and CTC workflow provides ease for biopharma clinical programs and maximize the value of priceless blood sample. One set of sample, two sets of answer. Here we show you the result of both genotypic and phenotypic analysis from a single blood draw. As of today, we partner with 9 out of the top 10 leading global pharma companies in ctDNA and CTC profiling. We have companion diagnostic program ongoing in US and China. To summarize, Predicine and Epic partner and create a global leadership in next-generation liquid biopsy. The future of liquid biopsy has come. It is ctDNA and CTC-based comprehensive cancer profiling with just one blood draw. With that, I would like to close today's presentation and encourage you to consider how this next-generation comprehensive ctDNA and CTC liquid biopsy solution may contribute to your important clinical study. Please contact Predicine for ctDNA analysis and contact Epic Science for CTC study. We thank you for your attention.